So the last class we were discussing about insecticide resistance. Today we will be dealing with a new topic, that is pesticide metabolism. That I think it can be completed within a single class. So today I tried to complete it. I took pesticide metabolism now because once you have studied resistance, pesticide metabolism is also somewhat related. Once you go through the class, you will understand it's somewhat related, because metabolism is nothing but it can be activation or detoxification. So if it is detoxification means, if it is an increased detoxification means, it will lead to insecticide resistance. So both way it is linked, resistance and detoxification. That's why I am taking up metabolism immediately after resistance. Okay, I will share the presentation. Okay. Okay. So any pesticide metabolism, or how a pesticide acts in an organism, it's a cascade of events. That means it starts from, in the case of, say, in the case of pesticides, before exerting its toxicity and after exerting its toxicity, it goes through a series of events. That's why it is called as a cascade of events. The first event is the absorption of the pesticide. If it is a contact poison, contact insecticide, it has to be absorbed through the cuticle. If it is a stomach poison, it has to be absorbed to the digestive system. So the first step in the toxicity of any pesticide is its absorption. If it is a respiratory poison, it has to be absorbed through the spiracles, through the respiratory system. So the first step in the metabolism, or the activity of a pesticide in a living organism, in an insect, is absorption of the pesticide. And second one is, once the pesticide is absorbed into the body, insect body, it has to be distributed in the insect body. Once it is distributed in the insect body, it will reach the target site. So once it is a nerve poison, say an organophosphate, it acts on the synapse. So once it is absorbed, the OP pesticide is absorbed, it has to be distributed and it should reach the synaptic junctions. Then only it can bind to acetylcholinesterase enzyme. So it has, once it is absorbed, it has to be distributed throughout the insect body. And once the distribution is completed, it will undergo metabolism. And this metabolism, I told you earlier, this metabolism can be either activation or detoxification. That's the first thing I told you regarding this class. So it undergoes the cascade of events. The first one is a series of events. First one is absorption of the pesticide into the insect body. Then second one is distribution. Third one is metabolism. Then distribution, I repeated it twice. Once it is distributed in the insect body, it will interact with the macromolecules in the insect body. It can interact with RNA, it can interact with the DNA, it can interact with protein. Interaction with proteins means, say, receptors, or say enzyme, say OP insecticide, which was the example we took. It can interact with acetylcholinesterase enzyme. Acetylcholinesterase enzyme is a protein. So once it is absorbed, it is distributed in the insect body, it is metabolized. Once it is distributed in the insect body, it will interact with the macromolecules present in the insect body, such as DNA, RNA, or protein. And once At the end of its interaction, at the end of its metabolism, it will be excreted. So the series of events that occur is the first step is absorption, followed by distribution, then metabolism, interaction with DNA, RNA, protein, and at the end it will be excreted. In between, if the insect gets killed, the excretion won't happen. So otherwise, it will be excreted. So this is the steps, series of steps by which a pesticide exerts its toxicity. And today we will be mainly dealing with detoxification. So because we are dealing with pesticide metabolism, we'll be dealing with activation also, a bit of it. But this detoxification can happen in two ways. The first one is phase 1 metabolism and the second one is phase 2 metabolism. It can be detoxification or activation both. Actually, it can be, should be, instead of detoxification, I should have given it as metabolism. Here, I will tell you. See, here, instead of detoxification, I should have given metabolism. The metabolism is divided into phase 1 metabolism and phase 2 metabolism. Phase 1 metabolism involves oxidation, reduction, and hydrolysis reactions, and phase 2 metabolism includes conjugation reactions. I will repeat, the metabolism of a pesticide is divided into two. One is phase 1 metabolism, and second one is phase 2 metabolism. Phase 1 metabolism includes oxidation, reduction, and hydrolysis. Phase 2 includes conjugation reaction. Is it clear till now? Now we'll move into each kind of metabolism. Yes or no? I am unable to hear you. Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, yes. Okay. Okay. So I'll just show you a graphical representation of how this happens. Uh, the first step is absorption. The insect will be exposed to the pesticide and the pesticide will be absorbed through the portals of entry. Portals of entry, it can be cuticle, it can be digestive system, and it can be pyracles. And once it is absorbed, it is distributed. Sometimes if the pesticide, if the insect is highly resistant, it can directly lead to excretion at that stage itself. But that is rare. From the portals of entry itself, if it excretion is very rare. But once it is distributed into the body, it will get activated. That is metabolism into more toxic metabolites. The second one is metabolism into less toxic metabolites. That is detoxification. The first one is activation. Second one is detoxification. That is metabolism to less toxic metabolites. And the third one is formation of conjugated products. And again, this metabolized products, it can be, say, malathion. Take the case of malathion. Malathion, it is distributed into organs. There it will be activated into malloxone. This malloxone is also distributed into the organ tissues. Sometimes, once it is distributed, it will directly interact with molecules. There won't be any detoxification, activation, or conjugation. If it is activated, it's okay, fine. It will interact with macromolecules. But it is detoxification or conjugation, it will reduce the toxicity of the pesticide. If if the pesticide is distributed into the insect body, and if it directly interacts with the protein, DNA receptors, etc., then it will greatly exert its toxicity without reduction in any toxicity. But if it goes through this step, detoxification and conjugation, then it will lead to the excretion of the pesticide, because once it is conjugated, it will become more hydrophilic. It can be easily excreted. This macromolecules, once it is interacted with the macromolecule, that is protein, DNA, RNA, etc., uh, it can lead to some damage to DNA, some damage to RNA, etc. Then it has to repair it, and the pesticide will be excreted. Once it interacts with the macromolecule, it exerts its toxic effects such as genetic, it can be genetic, carcinogenic, reproductive, neurotoxic, etc. Just a minute. Sorry. Uh, so, could you get how these steps happen? Whether this picture is clear? The absorption, then distribution, then metabolism. After the metabolism, it can be degraded project or product or activated product or conjugated. It can be again distributed into the in the different parts of the body, and the active molecule or the metabolized molecule can interact with the macromolecules RNA, DNA, and protein, and it will exert its toxic effect. In between, it can lead to, at all the steps, it can be excreted. If it is excreted in between steps before interacting with macromolecules, the toxicity of the pesticide will be decreased.
Sir,
Clear?
turnover,
In the
turnover and repair method.
turnover and repair, say one thing happen is, say if it interacts with the DNA, okay, some damage to DNA occurs. Then if some damage to DNA occur, it can be lead to it can have some carcinogenic effect. Mutations can. It has the system has its own repair, isn't it? You might have studied DNA repair of DNA. I have studied it in your biochemistry course, biotechnology course. Yes or no?
No.
No. Every time the DNA undergoes replication, uh, there can be a chances of some mutations happening during the replication, because replication is a constant process. Every time a cell divides, to lead to replication has to occur. So randomly some mutation can occur, and the body and the cell has its own repair mechanism to overcome this mutation. It will correct it, it will repair it. It's a almost all the organism have a repair mechanism on its own. So that means any damage to the macromolecule caused by the pesticide can be repaired by the uh, body itself. And the next one is turnover. The turnover is, say, acetylcholinesterase enzyme. Acetylcholinesterase enzyme will be inhibited by this OP insecticide. So that means this acetylcholinesterase will not be available for removing acetylcholine from the acetylcholine receptor. Acetylcholine is bound to acetylcholine receptor at the synapse, at the postsynaptic membrane. This acetylcholine has to be broken down into acetic acid and choline by acetylcholinesterase. But The acetylcholinesterase is inhibited by OP or carbamate insecticide. So the turnover of acetylcholinesterase enzyme is affected. Sometimes in resistant insects, what we have seen, it have gene amplification. That means it will produce more quantity of acetylcholinesterase. That is more turnover of acetylcholinesterase. Understood?
Yes, sir.
Yes. Okay. The, I will show you a second diagram where to describe phase one and phase two. This part, any xenobiotic, consider it as pesticide. Uh, pesticide, the primary thing that is chemical reaction that is happening during a pesticide metabolism is the conversion of a lipophilic molecule into hydrophilic molecule. Hydrophilic molecules can be easily excreted. So this is the pesticide. The oxidation, hydrolysis or reduction reactions happen and it will lead to the formation of primary products. This oxidation, hydrolysis or reduction reactions, these are phase one reactions. Oxidation, hydrolysis and reduction reactions are phase one reaction. And these primary products gets conjugated with compounds present in the body itself, such as sugars, amino acid, glutathione, phosphate, sulfate, etc. This materials, sugar, amino, all those things are present endogenously in the body itself. So this primary products gets conjugated with the molecules present in the insect body itself, and it will get excreted as it is hydrophilic. So this conjugation reactions are nothing but phase two reaction. So up to this it is phase one reaction. After this it is phase two reactions. Clear?
Yes, sir.
Okay. So let's see phase one reactions. Mostly the phase one reactions are carried out by cytochrome P450 monooxygenases. Sometimes other enzymes are also involved. There's mixed function oxidases or cytochrome P450 monooxygenases, the major enzyme involved in phase one reaction. The overall reaction, it is given over here. This is a substrate. Substrate, you always take it as a pesticide in our scenario, in our context. It is, we are dealing with pesticide metabolism. So always consider substrate as a pesticide. So the pesticide will interact with cytochrome by 450. Mainly it has a NADPH, it interacts with NADPH, and the substrate will get hydrolyzed. ROH will be converted to ROH. A addition of a hydroxyl group to the substrate will make the substrate or the pesticide more hydrophilic. OH group hydrophilic, COH group hydrophilic. So addition of a OH group. So this will make the compound more hydrophilic. Usually the enzyme catalyzes different kinds of reactions such as epoxidation, hydroxylation, N-dealkylation, O-dealkylation, desulfuration, sulfoxidation, etc. I will show what are all the reactions such as epoxidation, hydroxylation, N-dealkylation, etc. with examples in the next slides. And all these reactions such as hydroxylation, desulfuration, sulfoxidation, dealkylation, deaerylation, all this will lead to the addition of hydroxyl group, carboxyl group or epoxide group. Addition of hydroxyl group or carboxyl group, usually the compound will get detoxified because the addition of hydroxyl and carboxyl group makes the compound more hydrophilic and it can be excreted. Addition of epoxide group, normally the compound gets activated. We'll discuss it in the next slide with examples. So the phase one reaction, the first one is oxidation. We will see both oxidation, reduction and hydrolysis reactions. In the oxidation reaction, it is mainly catalyzed by cytochrome P450 monooxygenases. So it will lead to this oxidation will lead to epoxidation, hydroxylation, desulfuration, sulfoxidation, and dealkylation or dealkylation. It will lead to the addition of hydroxyl, carboxyl, or epoxide group. So this is the oxidation reactions that is happening in the phase 1 metabolism. So we'll see it one by one. What is N-dealkylation? N-dealkylation is nothing but the removal of an alkyl group from the nitro group or the amine group. C, phosphamidone, RnC2H5, C2H5. This is the amine group or nitro group. From that, this alkyl group C2H5 will be removed. So this is N-dealkylation. Removal of dealkylation means removal of alkyl group. Alkyl group will be removed from the nitrogen amine position. O-dealkylation, as we had discussed earlier, removal of alkyl group from the O position, from the oxygen atom. See, this C2H5 will be removed from this alkyl group, will be removed from the oxygen atom. That is O-dealkylation. Removal of alkyl group from the N position or the O position. So this is example phosphamidone to desethyl phosphamidone chlorphenvinose to desmethyl chlorphenvinose. The next one is epoxidation. That means addition of an oxygen atom. See it over. This is septachlor. We have a double bond over here, and at the position of double bond, there will be an epoxide. This is the formation of an epoxide molecule. Are you clear with these three? N-dealkylation, O-dealkylation, and epoxidation. These are just chemistry thing. Yes. Then ring hydroxylation. That means hydroxylation. That this is carbaryl. We'll add a hydroxyl group to it. That is ring hydroxylation. That means hydroxylation of a ring structure. Then side chain hydroxylation. That means instead of addition of hydroxyl group to the ring, here the hydroxyl group will be added to the side chain. COONH3. At the end of the CH3, we will add a hydroxyl group. Once hydroxyl group is added, even to the ring or the side chain, the compound will be get more hydrophilic. The next one is sulfoxidation. So here you can see this is phorate. This is a structure of phorate. We have two sulfur atoms over here. In the second sulfur atom here, we will be adding a sulfur group. The mixed function oxidases or cytochrome P450 monooxygenase will add a oxygen group to it, and it will become phorate sulfoxide. So we have seen N-dealkylation, O-dealkylation, epoxidation, ring hydroxylation, side chain hydroxylation, and sulfoxidation. The next one is desulfuration or dearylation. Desulfuration is nothing but the removal of sulfur group. Or dearylation is nothing but, what is an aryl group? Let me check how many of you remember your. Huh? What is an aryl group?
Benzene ring.
Yes, benzene ring. Anyone with the benzene ring? Okay. So here, yeah, you can see this is parathion. In parathion, it is converted to paraoxone by removal of this sulfur atom. So that means desulfuration. But this step, as you all know, is this activation of detoxification, removal of sulfur atom and addition of an oxygen atom. This desulfuration reaction. Activation or detoxification?
Detoxification.
Mm, I told you several times, malathion to malaoxone, parathion to paraoxone. What is it? Just a minute. What is it? Parathion to paraoxone, malathion to maloxone. What kind of an insecticide we call? Uh, uh, these are typical examples of Bindu, Swadi.
That's sir, it is activation reaction.
It is activation. So, what kind of insecticide is parathion?
Pro-insecticide.
Pro-insecticide. Yes. So here desulfuration is an activation step, while dearylation, that means the removal of this aryl group, this ring structure, that is a detoxification step. So we have seen several reaction: N-dealkylation or dealkylation, um, epoxidation, ring hydroxylation, side chain hydroxylation, desulfuration, dearylation. Then one more we have seen, I forgot, sulfoxidation. Okay. So this is in nutshell what we have discussed: epoxidation, sulfoxidation, that means addition of sulfur atom; desulfuration, sulfuration, removal of sulfur and addition of oxygen. All these three, epoxidation, sulfoxidation and desulfuration, these are activation reactions. And N-demethylation, that means removal of this methyl group. O demethylation or demethylation can be also be called as N-dealkylation or NO-dealkylation. Methyl group is an alkyl group, so you can write it as N-dealkylation or O-dealkylation. Removal of methyl group or alkyl group from nitrogen atom will lead to detoxification. Removal of alkyl group from oxygen atom will lead to detoxification. Then comes hydroxylation. That means addition of hydroxyl group. It is nothing but a detoxification reaction. This is in summary. Earlier I had given examples with pesticides. So are you clear? Till now we have discussed about phase 1 reaction. In the phase 1 reaction we were discussing about oxidation, mainly catalyzed by mixed function oxidase. Okay, shall I move to the next?
Yes, sir.
The next step is hydrolysis. Hydrolysis is, hello. എവിടെ അവിടെ റൈറ്റ് സൈഡിൽ കാണുന്നതല്ല, ലെഫ്റ്റ് സൈഡിൽ എന്ന് ഞാൻ പറഞ്ഞില്ല. ഇപ്പൊ അവിടെ ലെഫ്റ്റ് സൈഡിൽ അതിന്റെ ബോർഡിൽ എന്താ വേറെ ഉള്ള ക്ലൈമറ്റ് അല്ല, അവിടെ നിന്ന് ഫ്രണ്ടിലേക്ക് വന്നു. So hydrolysis, I told you several times this example. Any insecticide with ester linkage is susceptible to hydrolysis. So this one example, normal example. What is the largest group of insecticide with ester bond?
OP.
OP. Yes. Then can you name another group? OP, not
Synthetic.
all. Huh?
Synthetic
Synthetic
pyrogen.
Synthetic
pyrogen.
pyroxanes, yes. This ester bond is highly susceptible to hydrolysis, and esterases are the enzymes normally involved in the breaking down of the ester bond into the constituent acid and alcohol. As you studied in your chemistry in your school, acid and alcohol react to form esters, and these esters will be broken down by esterases into corresponding acids and alcohols. See, this is RCOR. This is the general structure of esterases. It will the esterases will catalyze the hydrolysis reaction by addition of this water molecule. It will lead to RCOH and ROH, acid and alcohol. So the major enzyme is esterases, carboxyl esterases. It also has enzymes such as phosphatases and hydrolases. So carboxyl esterase is a major group. It has two more enzymes in this involved in this hydrolysis reaction. The first other ones are phosphatases and hydrolases. See, malathion. As Bindu told, it is for OP insecticide also has ester bond. So this is COO. This is the ester bond over here. Another ester bond is also a COO. Say malathion. This COO bond is broken down into this acid, alpha monoacid C, and it will be broken down into alpha monoacid plus this will be ethanol. So this will be acid and alcohol. So it can happen it over this position. That means this ester bond or this ester bond. If it happens over this position, this is alpha monoacid, and if it happens over this position, it will be beta monoacid. Monoacid. In the same manner, it will happen into paraoxone also. This paraoxone, this is POO. This is phosphatases. It will be broken down into phosphoric acid and alcohol. Understood? Is it clear?
Yes, sir.
The next step is reduction. Reduction is very rare. Normally, hydrolysis and oxidation are the major phase one reactions happening in insects. Reduction is very rare. Usually, we have three types of reduction, that is nitro reduction, azo reduction, aldehyde or ketone reduction. Uh, this examples I have given. This is nothing but nitro reduction. You can see parathion, NO2 group. Uh, this to this NO2 group, you can see a hydrogen is added. Reduction is nothing but addition of hydrogen atom. You have to go back to your chemistry classes, isn't it? Addition of oxygen is oxidation. Addition of hydrogen is removal of oxygen, and addition of hydrogen is reduction. So this is nitro reduction. And here also you can see dehydrochlorination reaction, DDT. The one chlorine atom is removed and hydrogen atom is added. This is also reduction. But this kind of phase one reactions are very rare in insects. So among phase one reaction, we have discussed oxidation, hydrolysis, and reduction, of which oxidation is catalyzed by mixed function oxidases. Hydrolysis is catalyzed mainly by esterases. It also includes phosphatases also. And reduction, it is very rare in insects. Clear phase one reaction?
Yes, sir.
Yes. Just a minute before. Okay, coming to the next one, it is phase two reaction. Phase two reactions are nothing but conjugation reaction. I told you the pesticide molecule. Before going into that, can you name an enzyme which we studied earlier in insecticide resistance, which is involved in conjugation reaction? We discussed it several times. One enzyme which is involved in insecticide resistance is mainly involved in conjugation reaction.
You,
Louder. Bindu, louder. I can't hear you.
Uridine diphosphate.
Uridine diphosphate, yes, that is one conjugation reaction. One more,
ABC.
ABC. Actually, it's not a conjugation one. ATP binding acids are not, it's an excretion reaction. No. One thing, one enzyme I repeatedly told.
Glutathione transferase.
Yes, glutathione transferases. So the products of phase 1 reactions, after the phase 1 reaction, it will get conjugated with endogenous molecules. Endogenous molecules means molecules present in the insect body itself, such as sugars, amino acids, glutathione, phosphate, and sulfate. So it will conjugate with this endogenous molecule. Sugar is already present in insect body, amino acid present in insect body. And once this conjugation reaction happen, the compounds gets more hydrophilic and can be easily excreted. So that's what I told. It is more polar. More polar means more hydrophilic, more soluble in water. It becomes less toxic and readily excreted. And there are three conjugate types of conjugation reaction. The first one is type 1, type 2, and type 3. In type 1 conjugation reaction, it is activation reaction. Conjugating agent is activated. It's not, sorry, it's not an activation reaction. Sorry, I told it wrongly. In type 1 reaction, it is conjugating agent is activated. That means, say, glutathione S transferase. The glutathione S transferase is the enzyme which is involved in the transfer of reduced glutathione to the substrate. Here, the conjugating agent is reduced glutathione. I think Bindu was there. I forgot who told about uridine diphosphate glucosyl transferase. This uridine diphosphate glucosyl transferase. This is the enzyme which transfer uridine diphosphate glucose to the pesticide to conjugate with the pesticide. Here, the conjugating agent is uridine diphosphate glucose. So here the conjugating agent is activated. Substrate is not activated. That is, the pesticide is not activated. Say, if it is conjugating with sugar, the sugar will be activated. If the pesticide is conjugating with amino acid will be activated. If the pesticide is conjugating with glutathione, glutathione will be activated. So here the conjugating agent will be activated. Mostly it can be glucose sulfate or phosphate conjugation. We will see the example. In the type 2, reverse happen. The conjugation agent is not activated. Instead, the substrate, that is the pesticide, will be activated. That is type 2 conjugation reaction. The example is amino acid conjugation. And in the third type of conjugation reaction, both conjugating agent and the pesticide won't be activated. There won't be any activation reaction. Clear? Type 1, type 2, and type 3.
Yes, sir.
Yes. I will take the examples, then it will be more clear. See, this is glucose conjugation. This is example of type 1. That's what I told you, uridine diphosphate glucose. So this is glucose 1 phosphate. This one. It will it will react with UTP, uridine triphosphate. Uridine triphosphate contains energy just like ATP, adenosine triphosphate, uridine triphosphate. So uridine diphosphate glycosyl transferase is the enzyme. So this UDP will get transferred to glucose and it will form UDP glucose. UDP glucose means glucose will get activated. Glucose will get activated into a more energetic molecule. And this UDP glucose, see this UDP glucose reacts with the substrate or the pesticide. It is already hydrolyzed one after the phase 1 reaction. It is hydrolyzed. So that's why it is ROH. Substrate is having hydroxyl group. So this uridine diphosphate glucose, UDP glucose, will react with the product of primary or phase 1 metabolism, and it will form a more hydrophilic or more polar molecule. So here you can see the conjugating agent is glucose, sugar. The sugar is activated by the addition of UDP. UDP is an energy carrying molecule. Understood? How a conjugating agent is activated? Yes or no?
Yes, sir.
Yes. The next one is sulfate conjugation. Here, this is the sulfate molecule. This is ATP. ATP will react with sulfate. So the sulfate will form APS. APS, I have written it over here, adenosine phosphosulfate. This adenosine phosphosulfate again it reacts with another ATP molecule and it will form PAPS. PAPS is nothing but phosphoadenosine phosphosulfate. This phosphoadenosine addition of phosphorus to this ATP molecule will make it more energetic. So the conjugating agent is, that means the sulfate is the conjugating agent. Conjugating agent is activated into phosphoadenosine phosphosulfate and it will react with the substrate pesticide which is already undergone phase 1 metabolism and is hydroxylated. So this interact with the pesticide, the phosphoadenosine phosphosulfate, it will lead to formation of a more hydrophilic compound. The next one is phosphate conjugation. It's nothing but the equation ROH, the substrate reacts with ATP. ATP is nothing but a phosphate energy carrying molecule and it will get excreted. So we have seen glucose conjugation, sulfate conjugation, and phosphate conjugation. In all the three cases, the conjugating agent is activated, whereas the pesticide is not activated. In the type 2 pesticide is activated, not the conjugating agent. And here, glycine is the most common conjugating agent used. The conjugating agent in amino acid conjugation, mostly it is glycine. You can see the substrate over here. This is the pesticide. The pesticide after phase 1 reaction, addition of a COH group. This pesticide, detoxified pesticide in the first step will react with ATP and acetyl coenzyme A and this coenzyme A will be transferred to the substrate or the pesticide. Addition of this coenzyme A, as you all know, acetyl coenzyme A, energy containing compound. So the pesticide molecule will get activated and it will conjugate with glycine. Here the conjugating agent is glycine and it will lead to the formation of a hydrophilic compound. Clear? The type 2 conjugation reaction, amino acid conjugation.
Yes.
Yes. Then the last one is, this is type 3. I forgot to write type 3. Remember glutathione conjugation is type 3. Here there won't be any activation of the conjugating agent as well as the pesticide. This catalyzed by the enzyme is glutathione as transferase and the conjugating agent over here is reduced glutathione. The enzyme is glutathione as transferase. The conjugating agent is reduced glutathione and the conjugate, once the pesticide conjugates with reduced glutathione with the help of the enzyme glutathione as transferase, it will lead to the formation of mercapturic acid and mercapturic acid is easily excreted and during the transformation of this conjugate of glutathione with pesticide into mercapturic acid, there are several steps. The first step will have the loss of glutamic acid. In the second step, we will have the loss of glycine to form a cysteine conjugate, and the cysteine conjugate will get acetylated to mercapturic acid. I will do the stepwise reaction, then you can easily understand. See, this is the pesticide molecule. It will get conjugated with glutathione. The enzyme here will be glutathione S transferase, which will help in the conjugation of reduced glutathione with this pesticide. See, this happened. So This is R. You can see it over here. This is the pesticide, and this is the glutathione. And the first is the removal of glutamic acid. You can see from over here, this is glutamic acid. So this glutamic acid will be removed first from this conjugate. And the next step is the removal of glycine. You can see this is glycine. This glycine will be removed in the next step. See, glycine is removed and leads to the formation of a cysteine S conjugate. This is nothing but cysteine. Cysteine will get conjugated with pesticide. So this is cysteine S conjugate. And from this cysteine S conjugate, acetylization, deacetylization is removal of acetyl group. Acetylization is the addition of acetyl group. Addition of acetyl group. So here an acetyl group will be added. You can see CH3 group will be added and leads to the formation of mercapturic acid, which is easily excreted. So this is type 3 a reaction of type 3 metabolism in the phase 2 reaction. So I am finishing it over here. We have seen the pesticide metabolism. Actually, it's a cascade of events. First is absorption, distribution, then comes metabolism. This metabolized product can also be distributed, and at the end it will be excreted. It is classified into phase 1 and phase 2. In phase 1 we have oxidation, hydrolysis and reduction. Oxidation is catalyzed by mixed function oxidases. Hydrolysis mainly by esterases. Reduction is very rare in insect, azo reduction and all can happen. In phase 2 mainly it is conjugation reaction. Conjugation reaction the conjugates with endogenous molecules, sugar, amino acid, etc. It is classified into 3: type 1, type 2 and type 3. In type 1, the conjugating agent, that is say sugar, activated to UDP glucose, and it will conjugate with the pesticide. The type 2, the pesticide will get activated. That also example we have seen. In type 3, both pesticide as well as the conjugating agent won't be activated. Example we have seen glutathione conjugation. Clear? I am finishing it over here. Any clarifications you need?